I will explain over the next 15 slides what the FPA partner have in mind regarding this development of quantitative system toxicology approach to improve the understanding of the safety of new medicine. So I'm doing this presentation on behalf of all the FPA members who participate initially to the call definition. So before we start with the first slide, I just want to give a few words introduction about what is quantitative and system toxicology approach. So some of you, hopefully attending today, should be familiar with the field called quantitative and system pharmacology, which has emerged over the last decade as a new way to do PK/PD, broadly speaking. And when we speak about quantitative and system toxicology, basically there is three aspects here. The first one is quantitative, which means that we want to measure processes and outcome in complex systems quantitatively. Then the system itself is a kind of representation of how we can deal with complex systems. So we want to model the behavior of an entire complex system which deal with emergent properties of a system. A key thing is this system integrates the big data, but is not defined itself as a big data project. And it merges deterministic and statistical representation of a biological system. The last keyword, toxicology, I guess here I might not need to take a lot of time, but it's just a subset of all biological responses that are linked to an adverse or multiple adverse events. So the difference between quantitative system pharmacology and quantitative system toxicology, I would say, is the bandwidth of the approach. Quantitative and system pharmacology deal with one particular question in one particular system, for example, a target or a defined pathway. In toxicology, we probably will have to handle multiple targets and multiple pathways at the same time, which make it a bit more complicated. Due to this complexity, FPA understand that basically we need a public-private collaboration to have this undertake jointly. So basically, why it has to be public and private? First of all, we believe that there is a nice opportunity here to develop a novel collaborative framework of quantitative approach for predicting drug toxicity. And we believe it's a good time now due to multiple factors. First of all, we have an increasing availability of public and private, let's call it omic data set, where there are genomic, proteomic, metabolomics, so a lot of big data available. So also, we have seen over the past year development of modeling and bioinformatic approach to make more sense out of this omic data set. On the other hand, we have also seen a big development of the PBPK, so pharmacology-based pharmacokinetics, or PBPK-PD, standing for pharmacology-based pharmacokinetics pharmacodynamic model for organ injury. The second thing is we have a unique opportunity through this project to address the clinically relevant safety question using really another way to think about it. And the key aspect of the consortium would be to make sure that we have some proposals who have a clear scope to translate preclinical to clinical toxicity. And the last opportunity is to merge really complementary expertise to build such quantitative and system toxicology approach. The reason behind here is we believe that not any university, not any small or medium-sized enterprise, nor any big pharma or small biotech can handle such topic because the breadth of skill set is very wide. We need to have experimental scientists like toxicologists, physiologists, DMPK scientists, also a lot of computational scientists, being able to manage large data sets, to manage computational biology, system biology, and finally modeling and simulation. So if we move to the next slide, I will provide you the objective of the full project. And in total, we have identified 5 main objectives. On this first slide, I will describe briefly the objective 1 and 2. The first objective is really to develop an open for the consortium member at the beginning of the project or during the time frame of the project, focus and sustainable knowledge database to build this system toxicology model. Here, we want to make sure that basically this project is not about building a new database using IMI funding. This database has to be built in a sense that it has to be focused to feed the hybrid model that we have in mind. And the hybrid models that we speak about are models that should merge system network biology with more traditional PBPK and PD model. So all the knowledge, omics, disease model, PBPK model, have to be put into this database in order that they can be used and useful for the project. So that has to be very, very focused, and the proposal needs to reflect that. The second objective is to provide a clearer understanding of the translational confidence from non-clinical species to human. And here we would aspire to see applicants with human data on organ injury. FPI would have also some, and we'll describe that later on the need from applicant and what FPI will bring. But we still want to make sure we have enough human data on organ injury. The second thing is we need clearly to have a better understanding of the preservation of the network and toxicity pathway across species. If we move to the next slide, the last set of objectives here would describe how we can have impact with this approach. So basically, the objective 3 is support the key risk assessment decisions such as safety margin, clinical monitoring, or reversibility of adverse event using mechanistic and quantitative modeling. For those who are familiar with the field of modeling and simulation, what we aim at here is in the same spirit with what has been achieved in the past for drug-drug interaction field. So basically, currently with modeling and simulation, we are more and more able to predict drug-drug interaction with minimal input of in vitro data or minimal clinical study. So we aim, the ultimate objective of this project is hopefully to bring the same kind of level of possibility in the future and really to make the best use of modeling and simulation in the field of toxicology. The objective 4 here is much more technical. It's great to have a tool or framework, and I hope we will have this framework by the end of the project, but you need to make sure you have the methodology to both visualizing and analyzing your complex high content data to support this drug safety assessment. We mean also by that that the interface needs to be well designed. You can have the best modeling and simulation tool. If you are not able to communicate around it, nobody will understand what you want to do with it. So it will be also a very important part of the project because we believe that this, the outcome of this objective 4 will really enable us to get the buy-in on the tool toward the regulatory, for example, or toward the broad scientific community. And finally, we hope that at the end of the project, our science or our project would help to inform regulatory decision by providing evidence supporting the usefulness of doing this QST modeling to support the safety risk assessment. Once again, hopefully similar at some point to what is done today for drug-drug interaction. So when you think about a private-public partnership, you need to make sure that you define in the beginning what is pre-competitive. And here we have a couple of ideas why we believe this project is pre-competitive. First of all, and very important, pharma industry and I think also academia partner and the society itself believe that a better understanding and prediction of patient safety is ethics. You want to make sure that you bring new medicine into the clinic to the best of your knowledge to prevent any side effect on one hand, but also make sure that you can provide patients with maybe innovative medicine as a measured risk. And this is what is really pre-competitive. The best we do that together, the best both pharma industry, but also the academic community and the society will have some benefit. Second bullet point is the understanding of the prediction of this interspecies similarity or the expected observed difference would help us to really understand how this biological network across species contribute to difference in toxicity. I think we all understand that um, some effect on the mitochondria can lead to toxicity, some effect on some nerve pathway can lead to toxicity. But what is not so clear is how much in a preclinical species this pathway is important in comparison to what we can see in human. So all this knowledge, again, at the end of the day, is pre-competitive. And the last and not least, the data bank or the database that we will be doing, which is unique and curated data source to feed this quantitative and system toxicology, is again here per nature pre-competitive. When we will have all of this, each people can then make use of the database maybe in a different way, and that may be where the non-pre-competitive aspects start to come in. But from all the brainwidth of the initial aspect of the project, all of these points are truly pre-competitive. So what can we expect in terms of impact for the R&D process? So the first thing is classically, most or a lot of safety assessment is based on what we call no AL, so no observed adverse event level. What we aim here is to refine a little bit the paradigm and maybe improve the way we assess safety, and maybe also identifying novel translational safety biomarker to improve the clinical monitoring. We still have a lot of time difficulty to understand safety, which leads to some what we call safety cap in the clinic, meaning that you do see some safety, some adverse event in your preclinical model, but you don't really understand it, and more importantly, you cannot monitor it. And if you cannot monitor it, the agency will provide you an extra cap, which might limit the dose that you can use this clinic. And that could be particularly problematic if you believe you have a very nice drug who can save life of patients, but for which you have hard time to really move in some doses to test your concept. And we believe that this work could help define this novel biomarker, or define or qualify maybe some of these biomarkers to help at the end of the day define better the safety margin. Second point is, and it's linked to the first point I just described, we still do a lot of preclinical work, but sometimes the translation to the human, and especially in terms of risk and not adverse event, so being able to predict the probability of the clinical adverse event is still weak. And we believe that this approach gives a new way maybe, or new tool to improve the predictability here. We hope also to see that this QST paradigm should probably guide the design of some clinical trial. For example, it is well known that this adverse events are generally multifactorial, and some of them might be worsened in some patients. We believe that with this tool, we can probably simulate some clinical trial defining some key population with key attributes which would make them more prone or less prone to toxicity, and hopefully enable some better clinical trial on one end, and on the other end, may be able to test some risky hypotheses in patients where the risk that would take would be minimal because these patients do not have this other environmental or extra factor which would put their life at risk. The fourth bullet point is very important also in the term of 3R. So if we are successful, we can well predict that basically it should lead to reduction of animal usage in the preclinical phases. And finally, we aim to provide white papers or guidance to demonstrate that we can use this quantitative and system toxicology approach for the safety assessment for both industry and hopefully at the end of the project or during the project for the regulators. If we move to the next, so here it's just a suggestion of how we would think the project can run. So we define, let's say, 2 cross work package, which are drafted here on the upper part of the slide, a first work package which would be project management, which would have project oversight, communication, dissemination, and interaction with regulatory. A second work package which would be more the technology aspect, so what we call enabling technology. So here, it's where we need to have experts in statistic, in mechanistic modeling, in stochastic modeling, in knowledge management. So it's really all the basic technology which we would need to apply after across each work package defined organ specific, which are the one on the bottom, which are a work package on heart, on kidney, on gastrointestinal immuno, and liver. What we mean by that? As I said at the beginning, QST initially is a framework. So basically, we want to develop this tool and technology to really being able to merge against this stochastic approach to more deterministic or mechanistic modeling. But we don't want to do that for the sake of doing it. We want to apply that to some toxicology question. And again, toxicology is already very broad, and we try to restrict that to some key organs where the major, or let's say, where major findings occur, but where also FPR company have already some expertise or some data or knowledge to bring into it. So we will go a bit deeper later on what we expect on all of this work package. But here you would read on the topics text, we're really precise for each of these heart, kidney, gastro, immuno, and liver, what are really the expectation, what type of question we'd like to answer, what type of model we want to develop. And we try to define also in the call proposal what was really in scope and out of scope. Here, I might expect some question. I would recommend maybe to ask some of these questions at the end of the session. And also, as Hugh was mentioning, if you have more very specific questions, you are welcome to get in touch with Hugh, who will get back to us to provide you some answers. This architecture is a proposal. We let the freedom of the consortium applicant to propose maybe other architecture if they feel that it will address the objective and address the deliverable. It's just a recommendation, but it might be different as long as the main objective and deliverable of the project are covered in the suggested new architecture. So now let's move to the contribution of the applicant, what we expect really from the applicant of the consortium. So I think I already mentioned different times during this presentation that we acknowledge that QST is a broad proposal for novel modeling method. And we spent in FPR during the call definition process significant amount of time to try to scope the project as precise as possible regarding the different organ and some questions that the industry have that we want to be addressed with this project. But on the other hand, for sure, we did not define all deliverable because we want to have the input also from the consortium applicant and idea from the consortium applicant to really refine the project deliverable and really help us to build the final project plan. So the key things here again that we expect again the applicant to refine clearly the project deliverable. It can start by asking the question we want to address more clearly. So defining a clear tox question relating to an organ and then drafting more or less how we can provide answer to this question under the framework that we propose, and again, defining clear deliverable. The second thing is all the proposal need to provide a data-driven approach for modeling. So we don't want to spend a lot of time to generate new data. We want to make use of all existing data. And again, there is really a lot of existing data, either completely public or sometimes through the consortium, through IMI consortium or others, or also coming from FPR company. So for the FPR company, we will give you some indication of what kind of data we have, but we really expect the applicant to look first of all existing data, and the proposal should be around how to integrate this data to make these PBP, KPD, or let's say hybrid models that we aim at. The proposal might have also some proposal to generate new data, but this new data generation should come quite late in the project when a clear gap analysis has already been made, and we should not start by again generating tons of omics data when already all these data are generated and available elsewhere. The second thing, the last thing is the proposal have to focus on network biology or medicine application for sure. What we mean by that is when we think about organ toxicity, for sure we can maybe get access to models which are available in literature or maybe in some academia, model of liver injury upon, I don't know, an environmental stress factor like metal, heavy metal intake or whatever. That could be part of this application. But then the applicant should make sure that he make a clear link between the learning we can get for this specific model toward a more drug-induced liver injury model. So that has to have a link anyway to the pharmaceutical business. Now, if we go a bit more into the detail, what are the key expertise that we are looking at from the applicants, because we found them either only partly in FPR or not at all in the FPR. So the first thing is we want to have expertise in modeling, experience and expertise in modeling and simulation with a focus on organ injury and or degenerative disease. So here the key skill set is the ability to work with heterogeneous system data using a hybrid approach. Again, having really an understanding how we can integrate omics data, reduce the complexity of omic data, and make use of them into a more traditional, maybe PBPK framework. We ideally, applicant coming with some existing semi-mechanistic model of some of this toxicity related to the organ are sure more than welcome. Non-clinical and clinical data, as well as non-clinical and clinical expertise in the pathophysiology of organ injury of the relevant organ I mentioned in the previous slide. So again, liver, kidney, gastrointestinal/immune system, and heart are welcome. The third one, and maybe here could be in academia, but also in small biotech or dedicated IT organization, is expertise in data organization and more specifically data management. Here what we have in mind is, again, we have to deal with, let's call it big data, and I think it's sometimes kind of challenging how to make sense out of this big data, so how to fish in the big data world the data that makes sense for this particular topic. So company or academia having good idea how we can make these kind of things happen to convert the data into knowledge in the framework of this project are really welcome. As I mentioned already initially, we need to build on data, and we expect the applicant to come with proposal how to leverage knowledge or data from open source or consortia. I just give here few indications, but it's not prescriptive, it's just descriptive. We can think for the liver of maybe leveraging data from the MIPDD project. If we think about the disease model, we can think about the DD-MOR. If we think about more the drug aspect, we can think about drug matrix. Again, I'm not sure that the FPR members who put this call have also the complete and exhaustive understanding of all open source information available, but we expect the participant to make a clear link in their proposal with existing data and really leveraging existing data. And last but not least, we want to make sure that this project is a success at the end, and success means ability to collaborate effectively across disciplines, sectors, between public and private partners. So we want to encourage people who have the thinking to really make consortium with academia, but also small and middle-sized company or biotech to really have a broad representation of the scientific skill set that we are asking for. If we move to the next slide now, we will speak a bit more of what FPR will bring to the project. So that's what we call the in-kind contribution. So this in-kind contribution would be, first of all, key expertise in risk assessment for relevant organ toxicity, right? Because that's the business of FPR, to make sure that when we want to bring a drug to the market, we understand and we predict the risk pretty well. And also we will provide some existing data on this relevant organ. So we'll contribute both, and I would say mainly in terms of non-clinical data and expertise for the relevant organ. So we have existing non-clinical data set for organ injury for relevant organ. We can give some example, but we have some for liver, clearly, for kidney, for GI, and also for heart. So for all of these organs, we have already data set. We have also, or the in-kind contribution could well be on this non-clinical, new non-clinical data generation, which would be only made by design, which means that when the project will move, we will for sure identify gap in the data set, and FPI is willing to provide its technological platform and expertise to fill the gap. So it could be in the omic area, genomic, mainly, maybe metabolomic, PK experiment, safety experiment, in vitro, in vivo toxicology or toxicity. So all these functions we have pretty well in FPR. We will also contribute to existing non-clinical gen network model, again, for at least for kidney, liver, and heart. And we will also contribute to semi-mechanistic PK/PD model for GI toxicity. So all of this has been already clearly identified across FPR members who already sign up for the project. Where appropriate also, FPR is also involved in other approach, for example, aiming at characterizing novel cultural approach, so for example, organ on a chip or this kind of technology. So we have access to this technology, and we can provide data from this model, again, on unneeded basis, as long as this proves to have a specific value for the modeling. We quote here the stem cell population dynamic as an example, but it could be something else. So again, if in the consortium proposal there is this kind of idea, FPR can contribute here, and can refine, and that can be refined during the deliverable alignment when the consortium is set. And finally, FPR will take the lead on the project management and the project direction. Okay, now I hope to give you a bit of pitch of why you should hopefully apply for this call and make a proposal. For the academia, what we can think about is basically you would have access to industry resources in terms, and also risk assessment expertise. You will have access to genomic resources. You will have access to cooperative development environment for novel hybrids, what I'm mentioning several times, PB, PK, PD model. You will probably have access to some extent to either blinded or unblinded way on clinical and non-clinical data, as we were speaking previously. So basically what you might want to have access to in the past but cannot access due to the fact that it was in FPR, you can probably get access to. For regulatory scientists, again, here we really aim, or the vision is to be transformational for the safety assessment. Some of the people can well believe that we are trying to shoot on the moon, and I can understand that. But for me, if we are not visionary or want to be at least transformative, then we should not do an EMI public-private partnership. We can do that with the classical frame of our either industry or academia. For SMEs here, which stand for small and middle-sized enterprise, Again, I see here a huge opportunity for a forum to develop open source data set, and also on the methodological aspect, we can think about um, small company who might have, for example, a clear understanding on the framework, how to develop quantitative and system pharmacology or toxicology, and how, again, develop this method in a regulatory framework. At the end, we want to impact, hopefully, regulatory. And for patient and patient organization, even if I would say maybe in the call itself, um, I don't know yet if we need really patient organization, but at least what we need to pay attention to is during the time life of the project, we need to make sure that we communicate the benefit of this approach to patient organization in demonstrating them that we might have a chance to, again, make a great efficiency in advancing safe therapy, again, also providing maybe access to therapy that we cannot provide in a classical framework. So here, I would spend a bit of time to discuss some of the deliverable of the full project. And again, we cannot put in one slide all the deliverable which are in the call. So I would refer the potential applicant to really read the call, and you will have, again, more detailed deliverable. Here, I just present the 4 big buckets of deliverable, and under this big bucket of deliverable, again, in the call, you will have more granular definition of those. And we don't want also, it's very difficult for us to be focused and precise, but also not too prescriptive. We want to make sure that applicants come with their proposal as well to refine this deliverable. So here at the high level, there is 5 main deliverables which match the objective of the project. The first one is coming with compendium database of biological interaction that we were speaking about initially. And here I just mentioned the key feature we want to see. So it's a curated database. We have only good data. It's an searchable database. It's an easy to update database as soon as we get new data. And it's also an easy to integrate information source for the mechanistic modeling. So that's the key feature. In the text call, we provide a couple of ideas or sub-deliverables to guide how we believe we can get there. But again, it's pretty well open for the applicant to come with clear or more refined proposal. Then the second one would be an identification and mapping across species of this key biological network that reflect this biological response pathway associated with mechanism of toxicity. So what we think here about is really, again, we know some of the key password. Let's think about, I don't know, lipid disturbance. We can think about mitochondrial dysfunction. We have, this kind of biological response pathway that we know are associated with some mechanism of toxicity. The vision here is make sure that at least we are able to map what is similar across species. And when we say across species, is a species used generally in toxicology assessment, which are mainly rodent, rat, mouse, dog, monkey, and maybe to some extent rabbit, but at least rat, mouse, dog, monkey, in comparison to human, are what we have in mind. The third bullet point here is really the deliverable for the organ. So what we would like to see is that at the end of the project, we have really quantitative and mechanistic model of toxicity supporting risk assessment for specific toxicity liability that would be occurring in liver, heart, kidney, and GI immune system. I think in the core proposal, we are at least pretty clear for what we want to see for heart and, or let's say pretty focused, not clear, but pretty focused on what we want to see for heart and GI. It is true that it's a bit more complicated for liver and kidney in both, for different reasons. In liver, I mean, there were multiple initiatives over the past 10 or 20 years to better understand DILI, so drug-induced liver injury. We know that it's our topic. We know that we understand that probably with our approach, we might not probably give the solution to DILI. It would be great, but we are realistic. So we want maybe to to here find the driver or the main driver of DILI maybe with this network approach. And for kidney here, we believe that there is already much more to understand in terms of PBPK of a kidney. Kidney has multiple drug transporters, is a very complicated organ, and just in terms of disposition of drug, there is already a lot to understand here. And if we understand already pretty well the disposition, we might have a better understanding of the toxicity linked to the disposition. So here we can encourage also proposal in that regard. And again, leveraging what is already existing and not starting from scratch when initial model, like again from liver, are already existing. The last bullet is again more about this enabling tool, so develop open and innovative analytical tool. What we have in mind here, we expect to see at the end of the project new way to do modeling, new way to search data or to convert big data into knowledge input for modeling, and develop this user-friendly interface that basically in the future maybe people can take module of toxicity that would have been defined in the lifespan of this consortium and maybe link this module together to answer again very or more specific toxicity question. So now we move to the two last slide, and here we try to even specify a bit more what are we expecting for each target organ. So again, here it's still broad because I cannot describe in the minute allocated to this webinar more detail. So again, get back to the document to have more granularity. But overall, what is in scope for each of these organs is a PBPK model for progressive organ damage. So that's what we call the deterministic approach. We know the damage we want to model, and we build a PBPK model accordingly. The second thing is we want to incorporate the relevant omic data that describes the network perturbation in each organ, and that's what we call the stochastic approach. That would be also for all organs. Then we want to, what is in scope is also a knowledge-driven approach to modeling specific issues. Again, we leave some freedom to the applicant to come with a refined question for a particular organ, as long as this question can be substantiated by some knowledge which is available and it's in the framework of the modeling, we will accept that, or I mean, it's on the scope. And finally, again, everything which incorporates species difference, non-clinical to clinical, is on the scope. What is clearly out of scope for some specific organ? For the heart, specifically, applications that deal with QT prolongation are out of scope. We believe here that this aspect is already pretty well covered with either mechanistic model or modeling and simulation, or at least that the field is already too much advanced to have benefit inside of this proposal. What is also clearly out of scope is generating or proposing model to generate supervised signature approach to omic data. This has been part of other initiatives, and we don't want to replicate also here another initiative of derivating signature for omic data. And in the field of DILI, so drug-induced liver injury, if applicants want to propose something in that field, then we recommend the application to clearly lay out what is the value of this new proposal in the DILI world, in addition to all numerous existing initiatives like, again, MIP-DILI. There is tons of consortium who try to handle DILI. So here the recommendation is make clear how you leverage from existing, what would be the benefit of the current proposal in the frame of DILI, and make sure that the reviewer at the IMI can understand the benefit of the proposal according to our recommendation. Now, if you want, I will hand over to Arish, who is also part of the FPR company who put together the call. It will give you one example to hopefully at the end of this presentation fix some of the idea of what we have in mind for the GI immune toxicity framework. So Arish, if you're online, the floor is yours for this final slide.
Yeah, thanks, Loic. Yeah, again, so this is an illustrative example of how one would go about doing this quantitative systems toxicology modeling. This is specific to GI, but hopefully can illustrate some of the ideas we have in mind here. And again, the idea is we're not trying to be overly prescriptive here. Bear in mind this is an illustration. So again, how you would start here is by clearly defining the question. In this case, what we want to do is we want to quantitatively predict drug-induced acute and chronic GI toxicity in man. I'm going to focus a little bit on the acute bit here, where you would think of this as being oncology drugs disrupting cell proliferation differentiation in the intestine. So that's the question. So what you really want to do here is to predict adverse events in the clinic. You want to predict the things shown on the right there, which is, what fraction of patients show different levels of GI adverse events, grade 1, 2, 3, 4. So you want to predict that thing on the right, but starting with preclinical data on the left. So essentially what you could have is you could have rat GI pathology data as well as in vitro data. I apologize, there's a little image there that's not showing up for the in vitro data. So that image is essentially a mini gut, a three-dimensional in vitro system that you can use to profile compounds. So how do you go from the left, the preclinical, to the clinical? Well, you need to account for the biology of the system itself. So in this case, it's the GI. We know that in the intestine you have distinct regions. You have the crypts and the villi. In the crypts, those are the active regions of cell proliferation. So you have stem cells giving rise to daughter cells, multiple rounds of division, and then these daughter cells differentiate into the enterocytes that populate the villi. So that's the picture shown in the middle here. So that's a mechanistic model of intestinal cell dynamics that you can build. Then you can also go to the literature and ask, I'm going to characterize the homeostatic properties of the system, the properties that you need to understand even before you consider drug effects, and you can get parameters for how this homeostatic property changes between rat and man. What is the cell division rate? What's the differentiation rate in rat, and what is that in man? So once you have that in place, then you have the ability to address drug-induced effects. And you can do that either by starting with the in vivo data. You can fit your preclinical rat data and ask, what is my compound actually doing to, say, things like cell proliferation, differentiation? And then because you have the model now that accounts for species differences, you can translate to man. But you can also think of adding in another layer here. You can have a submodule that links the target biology, the pathway level effects, to changes in cell differentiation and proliferation. So you can build greater depth in a model like this. And how do you use in vitro data? Where you can use the in vitro data either to adjust potency across species. You can say, I'm going to have an in vitro rat system and an in vitro human system, get IC50s from there, and use that to scale results from preclinical to clinical. But you can also use your in vitro system to understand the target biology, right? So you can have your pathway level model being informed by the in vitro data. So that step two is the critical bit where you're building these models that help you translate from preclinical to clinical. And these models are accounting for things like cell dynamics, but also the pathway level effects. And then the last bit, step three, is essentially where you have a set of exemplar compounds and you calibrate your model and you validate it using those exemplar compounds. So again, you'd have both preclinical and clinical data for such compounds. So this is one example of GI toxicity where here we started in the middle with a model which was informed by our own understanding of the system. Now, how you do that, how you come up with a model structure may differ depending upon the different organs and the nature of the data that you have. So for instance, if you have a lot of omics data, you can think in terms of how can I build my model structure using the omics data? Or do I look at things like adverse outcome pathways to help me come up with a model structure? So those are the kind of questions that one needs to address depending upon the data availability for each organ. So I'll stop there. So hopefully that gave everybody a little bit of flavor as to the kinds of things that can be brought to bear here.
Thank you, Arish. And I hope that normally an example and a visualization of what we want to get at is probably more useful, I guess, than all the other slides that we try to explain. I hope that this example gives you a better picture of what we think is this call about. And now I think it's the time to take any specific question. And what I would say here, I might not have all answer of the question. I will share this session with Arish also. If some of the question cannot be addressed now online, we will make sure that the IMI2 take note about the question, and we will address then the question offline. We'll get back to you with an answer in any case.
Thank you both very much to Loic and Harish for a great presentation. So now, as you mentioned, it's time for questions and answers. I will briefly remind you how you can ask questions. The first way is in writing. You can use the question panel, type your text and click send. We will collect all the questions and then take them on a first come, first served basis. If you want to speak directly with Hugh, Loic or Harish, you can also use the yellow hand button. We will unmute you and then you're going to be able to speak with us directly. So while we wait for the very first questions to come in, let me remind you that there is, as Hugh mentioned before, a webinar scheduled to deal with IMI's rules and procedures. So this webinar is going to be featuring a science officer from the IMI program office as well as a person from our legal team. And it will explain all the rules and procedures involved in IMI applications as well as the IP policy of IMI. Something that is also worth repeating, although Hugh and Loic have already mentioned it, please contact the IMI program office for any questions you may have. We would like to ask you to refrain from trying to reach out to the FPR partners, as this may create issues of transparency, fairness and openness of the process for all the other candidates. Also, the presentation and the participant list of today's webinar will be published, the presentation will be published online and you will receive by email the participant list. The information about the call can be found on the website. I sent you a link to the webpage of the call 6 under IMI2 at the start of the webinar, and the deadline for the first stage of submission is the 12th of January 2016. So if anyone has any questions, this is the best moment, of course, to go ahead. There's nothing coming on now, so maybe we'll leave you just a few seconds to come up with your queries. So we have Gaurav Kumar that has raised his hand. Just 1 second, I will unmute you. Gaurav, you are now able to ask your question.
Hi, I have a question regarding the preclinical models in toxicity. So as you said that mainly rodents and rabbits are considered. Do you think that there are other species like zebrafish which are really, in terms of reducing animals and having the high throughput kind of toxicity output, can be a potential model can be proposed?
So there was a huge echo. Sorry for the line, and I had a hard time to understand the question. I don't know, Arish, did you get it clear? Because for me there was really a lot of echo.
Yeah, Loic, I think the question was around whether using data from things like zebrafish was going to be in scope.
Yeah, that's what's exactly the point.
Okay, so I would say, as we are not prescriptive about the source of data, if the applicant come with data that can fit with the objectives that, for example, I don't know, if with zebrafish data we believe that it could be useful to predict some liver toxicity or some of the kidney toxicity again, as long as you show how this data can be incorporated into the framework, I think it's acceptable in my opinion. But we just need to see that with not us, but in the proposal, it has to be clear how this data would be used to make the modeling.
Yeah, right.
But as long as the other partner that will be with you in the consortium have a clear proposal how to integrate that, how to make use of this data in a quantitative and system toxicology approach, I would not see any reason to reject those, in my opinion. Arish?
No, I agree, but I mean the key thing is it would be useful to demonstrate how such data from such models has been used in the past to translate to clinical effects. So from a scientific perspective, it may be useful to address that.
Thank you both very much. We now have another question by Paul Jennings, who is asking simply, what is the deadline of 12th of January for? So that is for the submission of short proposals. It's technically called stage 1 submission, and it's 12th of January at 5 o'clock Brussels time. We don't have any other question for the moment. Maybe we can wait a few seconds again, or maybe your presentations were too exhaustive and left no doubt.
Hopefully clear enough.
Yes, it was very clear, so thank you. And to the participants, as we've mentioned several times, if you think of some questions after the webinar, or once you've read the text and you have some questions, then don't hesitate to contact the program office, and we'll do our best to get the answers to you. Nothing else.
So maybe we're going to wait a few seconds more, and then if no other question comes in, there is one. So Lawrence Andrew Nelson is asking, how many partners in total can we have on a project, and what is the total budget we can apply for?
So if memory serves me right, the total budget that is available from IMI2 is €8 million. And the number of partners that you need in total, well, you have to read the call topic text. You have to then build your consortium in light of the objectives and deliverables that the call topic text lays out. And so that could be any number. You will be able to determine from the different areas that are mentioned and the different techniques that are required. You will be able to come to an assessment on the different expertise, and on the basis of that, roughly how many partners you will need. I would just like to remind you of the importance of avoiding redundancy. So if you just focus all of your consortium partners on one area or one objective, that will not be viewed well during the review against a consortium that is balanced, where all of the different objectives and all of the different expertise needed is included. So there's no yes, no, there's no concrete number that we can give you, and you have to make an assessment based upon the information laid out in the call topic text. The minimum requirement is 3 legal entities from 3 member states or associated countries. But when you read the text, you will see that you will obviously need more than 3 entities. I'm sorry not to be more precise, but it really is dependent on the call topic text and the expertise required.
Thank you, Hugh. We have a follow-up question. Laurence is saying, do we have to have a minimum number, say 2 or 3 SMEs for SMEs?
For SMEs, there is no minimum number of SMEs. Our rules are quite simple. We need a minimum of 3 legal entities. So that could be universities, research institutes, SMEs, patient organizations. The trick is to make sure that you have the right balance of expertise in your consortium to address all of the objectives. So there's no minimum requirement for SMEs. Of course, to have several SMEs on board would be good, but there's no requirement.
Okay, I think we don't have any other question coming in for the moment. If there is any other query that you would like to send to us, please do so by sending an email to info@imi.europa.eu. And we thank very much both Loic and Harish for the presentation. Thank you, Hugh. Thank you, everyone, for participating. We wish you the best of luck, and we look forward to receiving your proposals. Thank you very much, and goodbye.
Thank you.